Okay, plasma membrane will be there. So one ova is having, we are talking about oogenesis. Ova is having, if you are looking into an ova is having a nucleus and a plasma membrane will be here. And rather than the plasma membrane, there will be one envelope. This envelope is known as the primary envelope. One primary envelope will be there. Primary envelope will be there. And of course, after the primary envelope, there will be a secondary envelope. So a secondary envelope will be also there. So primary envelope will be there and a secondary envelope will be there. So if you're looking in the case of mammals, in the case of mammals means the organism which are having mammary gland. The primary envelope is known as zona pellucida. The name of the primary envelope is zona pellucida. Zona pellucida is the primary envelope in the case of mammals. Whereas in the case of sea urchin, that is a model organism in developmental biology. In the case of sea urchin, we have to study about the sea urchin in detail. In the case of sea urchin, the primary envelope is vitelline. The primary envelope is known as vitelline membrane or vitelline envelope. Vitelline envelope is a primary envelope in the case of sea urchin. When we are looking in the case of mammals, when we are looking in the case of mammals, the secondary envelope is known as cumulus layer. Cumulus layer is a secondary envelope in the case of mammals. And the cumulus layer is made up of ovarian follicle cells. The cumulus layer contains ovarian follicles. Ovarian follicle cells are present in the cumulus layer. That is the ovarian follicle cells that is coming from ovary. This is coming from ovary, ovarian follicle cells, which is coming along with the ova from the ovary that is forming the cumulus layer. That is a secondary envelope in the case of mammals. In the case of sea urchin, whereas in the case of sea urchin, the secondary envelope is known as egg jelly. The secondary envelope is known as egg jelly in the case of egg jelly in the case of sea urchin. Okay, that is clear. Why there is the envelope, the envelope, the speciality of envelope, they are species specific. Species specific. What do you mean by species specific? So suppose if it is an aquatic medium, in an aquatic medium, there will be several ovas, right? For example, if you're looking into a pond, you can find the ova of a frog, you can find the ova of a species, fish. Among the fishes itself, you can find the ova of many fishes. But the sperm have to come towards their particular egg only, right? So they will be species specific. Egg of ova will not bind with the egg of fish. Egg of a particular fish will not come bind with the egg of another sperm of another fish. Okay, they are species specific. And the envelope attract. The duty of envelope, they attract sperm. They also attract the sperm. They can attract the sperm towards the egg. They will attract the sperm towards the egg. That is another thing. They will attract the sperm towards the egg. That is a feature of the envelope. So primary envelope is there and secondary envelope is there. Looking into the egg, the egg inside the plasma membrane, you can see the cytoplasm. Cytoplasm will be there. There will be the cytoplasm. When we are looking into the cytoplasm, if it is egg, okay, if it is a egg and it is the nucleus, okay, the egg will be having a peripheral cytoplasm. Egg will be having a peripheral cytoplasm that is in the periphery. In the periphery, you can see the peripheral cytoplasm. This one is known as the peripheral cytoplasm. This is peripheral. In peripheral region, you can see the peripheral cytoplasm. Peripheral cytoplasm will be there. That is present in the peripheral region. That is peripheral cytoplasm. What is the specialty of the peripheral cytoplasm? The peripheral cytoplasm will be thick. They will be thick. And in the peripheral cytoplasm, you can see microtubule monomers. Microtubule, very well what is microtubule that we have already discussed. That is cytoskeletal element, the skeleton of a cell, that is a microtubule. Microtubule monomers. Microtubule monomers will be also present in the peripheral cytoplasm. This peripheral cytoplasm is known as cortical, the granules, the peripheral cytoplasm contains thick granule, right? They contain thick granule. And the thick granules are known as cortical granules. They are known as cortical granules. The thick granules are known as cortical granules. that is derived from cortical granules are derived from Golgi, Golgi apparatus. Golgi apparatus, the cortical granules are coming from the Golgi apparatus, and they contains, the cortical granule contains, they contains what they are containing, you know, they contains proteolytic enzyme. They contains proteolytic enzymes also. Proteolytic enzymes are present in this cortical granule. The cortical granule contains proteolytic enzymes also. Then there is the peripheral cytoplasm. Addition to the peripheral cytoplasm, you can see the inner cytoplasm. Inner cytoplasm will be also there. The inner cytoplasm is clear. Inner cytoplasm will be clear. You can see the inner cytoplasm, and the inner cytoplasm will be clear in the case of this is thick granular, whereas the inner cytoplasm is clear in the case of ova, egg. Whether it is clear? Are you here? Clear?
Yes, sir.
Okay. So we are moving forward. So we are talking here about the oogenesis. Okay, we can discuss about oogenesis in detail. Oogenesis, how it is happening. Oogenesis, no doubt at all, that starts from oogenesis starts from progenitor germ cells, PGCs. You know very well what is PGC. In the last class, we have discussed about all these things. That is progenitor germ cell. From the progenitor germ cells only, the oogenesis will start. Okay. So from progenitor germ cell, this is PGC. Okay, this is a progenitor germ cell. From the progenitor germ cells, there occurs mitosis. Due to the mitosis division, you know, more numbers of cells will be produced. 2n cell will be produced, and again they will divide. They will produce 2n cell. They will produce 2n cell. They will produce 2n cells again. So they will produce large number of 2n cell. When it is happening, you know, all these events are happening. All these events are happening during the fifth week or fifth month. During the fifth month of gestation. Fifth month of gestation. So when the kid is present inside mother's body, they will start the development of, or they will begin the development of ova. In the fifth month of gestation, they will start the development of this PGC. And you know, approximately 6 to 7 million, they will divide until it produce 6 to 7 million oogonia. They will produce the 6 to 7 million oogonia. These cells are known as oogonia. They will multiply, the PGC will multiply, and they will produce 60 to 7 millions of oogonia will be produced. And after producing 6 to 7 million oogonia, what will happen? You know, the proliferation, the further division, proliferation ceases. Proliferation will be ceases up to after the cells are multiplied up to 6 to 7 million, they will stop their division. During the last month of pregnancy, last month, that is during the 9th month, you know, during the last month of pregnancy, what will happen, you know, they reinitiate the division. So they reinitiate division when the embryo is reaching in the 9th week of pregnancy, they will start again their division. So till now, what was happening, you know, till now mitosis was happening, right? Till now mitosis was happening. But during the last month, what they will start, you know, they will start meiosis. So a 2n cell, so this is a 2n cell, each of the cells are 2n cell. This 2n cell will divide and that will start its division. But what happened, you know, they gets arrested at the meiosis get arrested. Meiosis cannot be completed. Meiosis will be get arrested at prophase. The division will be get arrested at prophase. In prophase, you know, the division will be stopped. So for a division to complete, prophase, metaphase, anaphase, and telophase should be there, but it will be get arrested during the prophase stage. In prophase, which stage they are getting arrested? You know, in diplotin stage. In diplotin stage of meiosis one. Diplotin stage of meiosis one. During the diplotin stage of meiosis one, they will be get arrested, and a cell will be produced at a primary arrest condition. A primary arrest condition, a cell is produced at a primary arrest condition. This is that cell. This can be called as, or this can be called as a cell. Okay, this is a cell. We'll discuss about names and all. Why a particular arrest is occurring? You know, the arrest is because arrest is due to the lack of, or due to the absence of luteinizing hormone. Luteinizing hormone will not be present in embryo because the embryo's endocrine organs are not at all developed. After the development, only the embryo will produce all the sex hormones and all. Due to the lack of the sex hormone, what will happen? You know, the embryo is getting arrested at which stage? Embryo is getting arrested at a prophase stage. More specifically saying, in the diplotin stage of prophase 1, the arrest is occurring. That is known as primary arrest. And now the cell is known as, you can call the cell as primary oocyte. This cell can be called as primary oocyte. You can call this cell as primary oocyte. This cell is known as primary oocyte now. So the primary oocyte will be present in the body of a baby till she is attaining puberty. Till she is obtaining puberty, you know, in her ovary, a number of primary oocyte will be present. A number of primary oocyte will be denatured and all. Anyhow, a lot of primary oocyte will be present in the body of a kid, in a female, when she is attaining her puberty. In puberty, we know that LH is present, LH luteinizing hormone is present. As LH is present, you know what happened? Primary arrest. As LH is present, primary arrest removed. The primary arrest will be removed as there is LH. As there is LH, the primary arrest will be removed. So what will happen? You know, the first phase of what will happen? First phase of meiosis, that is meiosis 1. First phase of meiosis is completed. Then the first phase of meiosis will be completed there. First phase of meiosis will be completed and form the resultant cell. We will call it as primary oocyte. We have called this one also primary oocyte, but this was not completed its division. Now it is completed its division. We can call it as primary oocyte. That can be called as primary oocyte. Whether it is clear? It is clear. So this cell, what will happen to the cell? Listen to me. The cell that is in primary arrest condition, that is in arrest. This is an arrest condition. From the arrest condition, what will happen? You know, they will divide and they will produce two cells. One cell will be small and one cell will be large. The large cell is known as primary oocyte. This can be called as primary oocyte. And the small cell is known as first polar body. This can be called as first polar body. This one is first polar body and this one is primary oocyte. What will happen to the first polar body? The first polar body will again divide. First polar body will again divide and it will produce again polar bodies. It will produce polar bodies again. The first polar body will again divide and they will produce polar body again. What will happen to the primary oocyte? What will happen to the primary oocyte? You know, it is haploid now, right? It is haploid and it is haploid. The primary oocyte, they will increase in their size. Primary oocyte increase in size, okay? They increase in size. They will increase their size. Okay, they increase in size. How they are increasing size? You know, by producing, they will produce by producing storage molecules. By producing storage molecules, a lot of storage molecules will be produced and they will increase their size. Now it is a large primary oocyte. It is a large primary oocyte now. I hope it is clear. It is clear? Clear?
Yes, sir.
Okay. Okay. So what will happen to the large oocyte now? So a large oocyte is there. This is a large oocyte. This is a haploid. This is haploid in chromosome number. So this one is enlarged. That is bigger. Enlarged primary oocyte is this one. So what will happen to the enlarged primary oocyte then? This enlarged primary oocyte, they will undergo the meiotic division. So there will be the meiosis 2. Meiosis 2 will happen. The meiosis 2. When it is reaching in meiosis 2, what will happen? It is reaching in meiosis 2 division, it will continue its meiosis 2, but it reaches the metaphase stage. They reaches the metaphase. Whenever they reaches the metaphase stage, whenever they reaches the metaphase stage, what will happen? Get arrested. Again, they get arrested. They will start their second division. In second division also, they will get arrested at metaphase stage. They will get arrested. Okay. So what will happen? The arrested oocyte, actually, this condition can called as the secondary arrest. This is known as the secondary arrest. This is the secondary arrest. We can call it as the secondary arrest. Okay. So in a female, when a female is ovulating, when a female is ovulating, secondary, every month a female will produce one ova, right? So whenever a female is ovulating, secondary oocyte, whenever a female is ovulating, what will happen? The arrested oocyte, you can call it as secondary oocyte, or you can call it as secondary oocyte, okay? So the secondary oocyte, that is only secondary oocyte is, or secondary arrested oocyte. Secondary arrested oocyte is ovulated. So who is ovulated? Every month a female is ovulating secondary oocyte. She is only ovulating secondary oocyte. That secondary oocyte is not completed its division. So what will happen? What will happen? This is at large cell. This is at large cell. And this large cell, that is arrested cell. This is the arrested cell, right? This one is the arrested cell. This arrested cell, what will happen? Finds a sperm. This arrested cell that finds a sperm, and arrest is removed again. Arrest is removed only when this cell is finding an ova. So this cell, as it was getting divided, it was containing its polar body here. As it was dividing, it will be having a polar body here. So what will happen? Whenever the arrest is removed, polar body is released. Polar body is released out. Polar body will be released out. Whenever the arrest is removed, that will produce what? Whenever the arrest is removed, that will produce a small polar body. It will produce again a polar body, and this one is known as ova. This is known as the ova. So a female can produce only if, I'm asking a question to you, whether you are producing ova or not. Whether you all are producing ova or not. Whether you all are producing ova or not. Yes or no? Amina.
Yes, sir.
Whether you are producing ova every month?
Yes, sir.
You are producing ova?
Yes, sir.
Arul Jodi?
Sir, no, sir.
Arul Jodi is not producing ova every month. How can you, Amina, you can produce ova every month? You are not producing ova every month. Amina, are you married, Amina?
No, sir.
Then you can't produce ova, okay? Ova, that, okay. Arul Jodi, please explain that. Why you are telling that you are not producing ova? Arul?
Sir?
Yes, come on, tell me. Why we are not producing, why an unmarried woman is not producing ova?
Sir, sperm one irukadu.
Sperm should reach there. right? Sperm should reach there. Whenever the sperm is reaching near the ova, then only the secondary arrest will be removed. Then only that cell will divide and produce ova, right? Only if there is an entry of ova, then only the ova will be produced in female's body. Otherwise, a female body will produce what? Otherwise, a female body will produce only So what you are releasing every month, whether it is ova or secondary oocyte?
Secondary.
Secondary oocyte. You all are releasing secondary oocyte only. Whenever the entry of sperm is there, then only the secondary RS will be removed. Then only ova can be produced. Amina, are you producing?
No, sir.
Don't produce, okay, before marriage. Okay. Whether that point is clear?
Yes, sir.
Okay. So now we have completed the discussion how the ova is getting produced in a female's body. So normally a female is releasing secondary oocyte. Secondary oocyte can be converted into ova only if the secondary arrest is removed from that ova. Then secondary oocyte. Then only the secondary oocyte will be get converted into ova. Okay. Right. So that is clear, right? That is clear. So in oogenesis, there is, along with the oogenesis, one more phenomena is happening, that is follicular maturation. Follicular maturation is happening. Follicular maturation is also happening along with the oogenesis. So what is follicular maturation? We can look what is follicular maturation. So when it is primary oocyte, okay, when it is primary oocyte, I hope you remember when the primary oocyte is getting produced in a female's body. When it is primary oocyte, over the primary oocyte, a layer of granulosa cell will form. A layer of granulosa cell will form. Over the primary oocyte, a layer of granulosa, you can very well see a layer of granulosa cell will be there around the primary oocyte. So what is the granulosa cell? The granulosa cell means ovarian follicle cells. Ovarian follicle cells. This is the ovarian follicle cells. Ovarian follicle cells from ovary. Ovarian follicle cells from ovary. So now it is known as, you can call it as, now it is known as the total cell, the primary oocyte plus over the primary oocyte a granulosa there. Now you can call it as primary follicle. This one can be called as primary follicle. So during embryonic stage itself, the primary follicle will be produced in a female's body. So the primary follicle, what will happen to the primary follicle? This is a primary follicle. You are having the primary follicle. Above this, there is the granulosa layer is there. This one is a granulosa layer. This is a granulosa layer. Over the granulosa layer, more granulosa will be formed. Over the granulosa layer, more layers of granulosa will form. That is, granulosa proliferate. The granulosa proliferate well. Granulosa proliferate. The granulosa will be proliferating well. The granulosa will proliferate and it will begin to produce thecal cells around it. Around it, another cell will also form. This cell layer is known as thecal cell. Now you can call this one as, whenever the theca cells are coming, you can call it as secondary follicle. Inside the secondary follicle, you know very well, inside the secondary follicle, who is there? Our primary oocyte is there. Our primary oocyte is very well there inside the secondary follicle. Inside the secondary follicle, primary oocyte, over the primary oocyte, granulosa layer is there, and above that you can see the thecal cells also, you can see there. Okay. Now what happens? You can look. The granulosa began to produce. What they will begin, they began to produce cavity. In granulosa. In granulosa, they will begin to produce cavity. that cavity can be called as, such a cavity can be called as, for example, if you are looking, it will be in this manner. Our primary oocyte will be there, and the granulosa layer will be surrounding, there will be the granulosa layer, and the granulosa layer will grow in this manner, and they will also form cavity there. In this manner, they will form cavity also, and over this you can see, what? Over this you can see the theca also present there. This is a theca. This cavity can be called as andrum. This is a cavity, and the cavity is known as andrum. What is the specialty of andrum? Andrum will be filled with, proteinaceous fluid. Proteinaceous fluid will be present inside the andrum. Protein digesting fluid, that is proteinaceous fluid, that will be present in the andrum. Okay. Then the granulosa the granulosa cells, granulosa cells plus theca. Granulosa cells and theca together, what they will do, they produce estrogen. You know very well what is estrogen. That is female sex hormone. They can produce estrogen. So what happens next? In this, again, what will happen, the size of andrum, after this, what will happen? Size of andrum gets enlarged. Whenever the size of andrum gets enlarged, the size of andrum will get enlarged, and what will happen as a result, and push the oocyte. They will push the oocyte towards the periphery. They push the oocyte towards the periphery. What is that term indicating? So, this is the primary oocyte. Whenever the size of the andrum is increasing, whenever the size of the andrum is increasing, so the size of andrum is increased, and they pushes the oocyte towards the periphery. So now the oocyte will be present towards the periphery. This is a thecal layer, and now the oocyte is present towards the periphery. Now it is present in the periphery, isn't it? It is okay. So, if you are looking, now it is known as, at this stage, you can call this stage you can call it as tertiary follicle. Now. It was primary follicle, then it is secondary follicle, now it is tertiary follicle. You can call it as tertiary follicle. If you are looking in this stage, in tertiary follicle, if you are looking in this tertiary follicle stage, big andrum is present, granulosa layer is present, and theca, large amount of theca is also present. If you are looking into the oocyte now, this is a oocyte, right? When we are looking into the oocyte now, what the oocyte will be having? Oocyte will be having a plasma membrane. And it will be having the zona pellucida. We have already told about the zona pellucida. I hope you remember the envelope, that is zona pellucida. Plasma membrane will be there inside, and zona pellucida will be also there. Then, when it is becoming another stage, when it is growing further, that will become as, when it is growing further, they will become as the another follicle. When it is growing further, when all these structures are growing further, when the theca is arranged into theca externa and interna, even theca layer will be more here. Now another structure will be formed, and this one you can call it as the Graafian follicle. Now, this one is Graafian follicle. So in Graafian follicle, if you are looking, when it is in Graafian follicle, this one secondary oocyte, this is now secondary oocyte, that is in the oocyte is now in what, now in secondary arrest stage. The oocyte is now in secondary arrest stage. In secondary arrest stage means it is ready to ovulate, right? It is ready to ovulate. It is in secondary arrest stage. That means it is ready to ovulate. So who is getting ovulated? Who is getting ovulated? Who is getting ovulated? The Graafian follicle will rupture. Graafian follicle will rupture. It will break down, and who will come out? The plasma membrane. This is the ova. Ova is having plasma membrane. We know very well. And above that there is a zona pellucida. Zona pellucida is there. And above that, there is a follicular cells will be also there. This is a granulosa cell. This one is granulosa cell. A single layer of granulosa will also come out. In this manner only, the secondary oocyte is getting released. So this is, we are calling it as cumulus oocyte complex. We are calling it as cumulus oocyte complex. Who is getting ovulated? Cumulus oocyte complex is getting ovulated. Not only the secondary oocyte is coming out, along with the secondary oocyte, a single layer of granulosa will also come out. A single layer of granulosa will be other surrounding this, and totally you can call it as cumulus oocyte complex. It can call as cumulus oocyte complex. Okay, whether it is clear?
Yes, sir.
Okay. So when we are looking, the ova is released in this manner. Ova, its ova's nucleus, ova's plasma membrane will be there, and by surrounding that, you can see a zona pellucida layer. Zona pellucida layer is there, and a layer of granulosa will be also there. A layer of granulosa in this manner, a layer of granulosa will be also present there. The layer of granulosa appear like a crown. It will be appearing like a crown. So it is known as corona radiata. You can call it as corona. Corona, the virus name is coming from its own appearance, right? That is crown-like appearance. Anyhow, it is known as corona radiata. You can call it as corona radiata. This structure can be called as corona radiata. So it is coming from after a rupture, right? It is coming after a rupture of whom? A rupture of Graafian follicle, right? Graafian follicle ruptured, and from Graafian follicle only this came out. So the Graafian follicle, what will happen to the Graafian follicle? The Graafian follicle after ovulation, after the secondary oocyte is released out, the Graafian follicle will be converted into corpus luteum. Graafian follicle will be converted into corpus luteum. It will be converted into corpus luteum. It will become as corpus luteum. What is the role of the corpus luteum? The corpus luteum only secretes progesterone. Corpus luteum only secretes progesterone. They will secrete progesterone. Who is progesterone? Progesterone is pregnancy maintaining hormone. Progesterone is pregnancy maintaining hormone. So the pregnancy maintaining hormone is coming from the corpus luteum. If there is no fertilization, suppose no fertilization, there is no mating, okay, there will not be any fertilization. What will happen? The corpus luteum gets converted into corpus albicans. Corpus luteum will become as corpus albicans. Corpus luteum will be converting into corpus albicans and gets degenerated. Corpus albicans and degenerated in the absence of fertilization. If there is no fertilization, corpus luteum will become as corpus albicans and it will regenerate. Whether it is clear? Hello? Clear or not?
Yes, sir.
Okay. Listen for the next step. Okay. So we are going to discuss about its own hormonal control, how the hormonal control is happening here, that we are discussing here, hormonal control behind this. What is the hormonal control? Okay. So, everything will be coming from the hypothalamus, no doubt at all. Hypothalamus will be responsible for all these things from the hypothalamus, as like spermatogenesis, GnRH is coming. Gonadotropin releasing hormone is coming. The gonadotropin releasing hormone will act on anterior pituitary gland. It will work on anterior pituitary gland. Then the anterior pituitary will produce two pituitary hormone, that is follicle stimulating hormone FSH and the luteinizing hormone LH. FSH and LH is coming. FSH and LH, they are acting on where? FSH and LH, they are acting on ovary. They are acting on ovary. They will work on ovary. What ovary will do? Ovary will do low levels of estrogen. Ovary will secrete low levels of estrogen. It will produce low levels of estrogen. Whenever the estrogen is coming, the estrogen will go and the estrogen will inhibit the hypothalamus. For what? Already we are producing sex hormone, so no more signal is required for us. That information will be going from the estrogen towards the hypothalamus. The ovary will also produce inhibin. From ovary, inhibin will also produce inhibin. Inhibin act as negative feedback mechanism, and the inhibin will check. Inhibin go and check where, the inhibin check the anterior pituitary. Anterior pituitary will be checked. In a female, if you're looking in the case of a female, only 2 million cells undergo meiosis. Only 2 million cells will undergo meiosis and produce what? And produce primary oocyte. So in the life cycle of a female, the primary oocyte will again degenerate. The primary oocyte will again degenerate from birth to puberty. From a female getting birthed and she is attaining puberty, a number of primary oocyte will be degenerated. In a life cycle of a female, do you have any idea what will be the number of ova a female can produce during her lifetime? Any guess? The number of ova a female can produce during her lifetime? What is the number of sperm a human male can produce in his lifetime? What is the number of sperm? We have discussed in last class. What is the number of sperm a male can produce in his lifetime? Huh? Hello? Are you here? Can you hear me?
Yes, sir.
Okay. So a male during his lifetime, a male can produce millions of cells, right? Millions of sperms will be produced by a male during his lifetime. Whereas in the case of a female, a female, she can produce only 400 ova in her lifetime. In a lifetime, a female can produce only 400 ovas. Only 400 ova will be coming from a female's body during her what? During her life cycle after attaining puberty. That means in between menarche and menopause. Menarche means first menses and menopause means last menses. In between menarche and menopause, a female can produce only 400 ovas. That is also clear. If you're looking into another thing, we know there is a particular cycle is occurring in female that is known as menstrual cycle. That is occurring in a female's body, menstrual cycle. We are simply looking to the hormones present in the menstrual cycle. What all are the hormones present in menstrual cycle? We can look. So menstrual phase, we can classify the menstrual phase into different stages. From 0 to 5 days, there is a cycle. From 5 to 14, there is another phenomena. From 14 to 28, there is another phenomena. So, in a female's menstrual cycle, the first phase is known as, in a month, 0 to 5 days, that is known as menstrual phase. That time period is known as menstrual phase. During the menstrual phase only, a female will bleed blood. That is menstrual phase. From 5 to 14, that is known as follicular phase. From 5 to 14, that is known as follicular phase. All the follicles that we have discussed now is produced during this phase. That is during follicular phase. And after that, the last phase is known as luteal phase. This can be called as luteal phase. Corpus luteum is present during this phase. This is luteal phase. So, as we have already studied, during follicular phase, during this follicular phase, follicles producing what? Follicles are producing, we have already discussed that follicles are producing estrogen. So during follicular phase, what will happen? The level of estrogen will be high during follicular phase. The 14th day only, the 14th day, this is a 14th day. The 14th day, we will call it as ovulatory phase. 14th day is known as ovulatory phase. In ovulatory phase, what will happen? That rupture is occurring, graphene follicle is rupturing, and ova is releasing in the 14th day. So if you are looking to the count of estrogen, the level of estrogen will be high. The level of estrogen will be high. And when it is reaching towards the 14th day, the level of estrogen will become low because graphene follicle is rupturing. The growing follicles are producing estrogen. We have discussed in previous slides and all. Growing follicles were producing estrogen. So during follicular phase, the level of estrogen will become high. When it is getting ruptured, the level of estrogen will fall. This is happening during the follicular phase for estrogen. This is the first one. Okay, this is a level of estrogen hormone. This is a level of estrogen hormone. What will happen to progesterone hormone? The level of progesterone hormone will be minimal. There will not be any progesterone hormone till follicular phase. But we know after follicular phase, whenever the graphene follicle rupture, we have studied that. If you're just turning your page, there will be note. When the graphene follicle rupture, what will happen? The graphene follicle will become corpus luteum, and corpus luteum will produce progesterone. So the level of progesterone will be high in this manner. This is a graph of estrogen and progesterone in menstrual cycle. In the same manner, if you are looking for the level of follicle stimulating hormone, if you are looking for the level of FSH, the level of follicle stimulating hormone, FSH, follicle stimulating hormones are produced high in follicular phase. So in follicular phase, there will be high amount of follicle stimulating hormone, and its graph will be going in this manner. Another hormone, that is LH. Another hormone is LH, luteinizing hormone. The level of luteinizing hormone will be very high in 14th day. In 14th day, very high of LH will be there. That 14th day is known as LH surge. That 14th day is known as LH surge. This LH surge is only causing the rupture of graphene follicle. We know very well what was graphene follicle. In this manner only we were having this graphene follicle has to be ruptured, then only our secondary follicle will come. In the andrum, when we are looking in the andrum, we could find in the andrum there were proteolytic enzymes. In the andrum there were proteolytic enzymes. Proteolytic enzymes were present in the andrum, right? That were present in the andrum. So what is the role of LH? LH activates, LH only activating, LH activates proteolytic enzyme. LH activates proteolytic enzyme. LH will activate proteolytic enzyme. Whenever the LH activates proteolytic enzyme, what will happen? The graphene follicle will rupture and the ova will be released out. So a menstrual cycle is having 28 days. A menstrual cycle is having 28 days. Okay, correct. This is a menstrual cycle. Any doubt is there?
No, sir.
No,
Any doubt?
No, sir.
Clear? Okay. You're making notes, I think. Okay. Should be notes with you. Okay. Then we can look what are the uterine changes. If you're looking into the uterus, okay, if you're looking at a female's uterus, this is a female's uterus, suppose in this manner, this is a uterus, okay, this one is a uterus. Okay. This is a uterus region. This one is a uterus region. Okay. This one is a uterus region. Uterus in this one. Okay, uterus. This is uterus. This one is known as uterus. This is uterus. This is known as fallopian tube. This tube is known as fallopian tube. This tube can be called as fallopian tube. The first part of the fallopian tube is known as isthmus. This portion is known as isthmus. You can call this portion as isthmus. And you can call this portion as ampulla. This portion can be called as ampulla. This is an ampulla region. And this portion is known as infundibulum. This portion can be called as infundibulum. This is an infundibulum. Infundibulum contains finger-like projection. These projections are known as fimbriae. These projections are known as fimbriae. This is fimbriae. And here will be the ovary. This is the ovary. From ovary, ova will be released out. In ampulla only, fertilization takes place. Fertilization takes place here. Fertilization takes place in the ampulla region. Ampulla region, fertilization will take place. Okay. So, where the ova is released, the ovary will release ova into the body. Ova is not released into the fallopian tube. Ova is released into the peritoneal cavity. Peritoneal cavity. Where is a peritoneal cavity? Peritoneal cavity means there is a cavity inside our body. So, what will happen, who will catches this one, fimbriae. Fimbriae is a structure. Fimbriae catches it. Fimbriae catches it and help for its movement. So that want to move into the fallopian tube. Movement into the fallopian tube. Into the fallopian tube, the ova want to move. So the ova will be released into the peritoneal cavity. This one is the ova. Fimbriae will capture the ova. Fimbriae will capture the ova. Who will capture the ova? That is fimbriae. Fimbriae will capture the ova. Okay. So that will reach us into the ampulla. It will be reaches into the ampulla. So the conception happening period can be called as conception happening period is known as fertile period. That is known as fertile period. Fertile period means even if a male and female is getting mated, every mating will not lead to pregnancy. There is a particular time period. That time period is known as fertile period. During fertile period only a female can be impregnated. During that time period only a female can be impregnated. So what will happen after a ova reaches here, ova will wait 12 to 24 hours. 12 to 24 hours means 1 day. After 12 to 24 hours, what will happen, ova degrades. Ova degrades if no fertilization. If there is no fertilization, the ova will be degrading after 24 hours. Till 24 hours, the ova will wait, and after that, the ova will degrade if there is no fertilization. 48 hours, a sperm can wait. 48 hours is a sperm lifespan. A sperm can live up to 48 hours. 48 hours only, that means 2 days. 2 days a sperm can live, but inside female tract, inside female reproductive tract, it can live up to 5 days. 5 days. It can live up to 5 days. Up to 5 days, a sperm can wait. So up to 5 days, a sperm can wait, and the ova will be waiting. The ova will be waiting for what? The ova will be waiting for 24 hours, and the sperm can even wait for 5 days for the arrival of sperm. Okay. So this is a 14th day. Okay, this is a 14th to 15th day. There we are expecting ovulation. So a female is ovulating during her 14th to 15th day of ovulation. So if you are looking, if you are looking towards this, if it is a 10th day, if it is a 10th day, 10th day of her menstrual cycle, if sperm is coming, 10th day of menstrual cycle, if sperm is coming, the sperm will wait for 5 days, right? So conception can happen, pregnancy can happen. And ova will leave up to 2 days, right? Ova will leave up to 2 days. That means 17th day, right? Up to 17th day, there is a chance till 17th day. So from 10 to 17th day, during her menstrual cycle, from 10 to 17th day, in between 10 to 17th day, any time is risky period or any time is fertile period. So if you're looking for a cyclical representation of the menstrual cycle, okay, this is a menstrual cycle of a female, okay, this is a menstrual cycle. Cycle days starting from 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 20, 22, 24, 26, 28. This is a menstrual cycle. During 14th day, what is happening? We know during 14th day only. During this 14th day, ovulation is happening. During the 14th day, ova is coming out. Ovulation is happening. From 1 to 5 days, 1 to 5 days, we know very well that 1 to 5 days, the menstrual phase, bleeding is occurring. 1 to 5 days is a menstrual phase. That means bleeding is occurring during this phase. Then 5 to 14th day, what is happening? 5 to 14th day, that is follicular phase. Follicular phase, follicle is present there. That is follicular phase. After 14th day, that means 15 to 28, that time period is known as luteal phase. Corpus luteum is present. This is known as luteal phase. So if female is fertile during 10 to 17th day, 10 to 17th day, a female is fertile. 10 to 17th day, a female is fertile. We know 10 to 17th day, a female is fertile. So this time period can be called as the fertile period. You can call this as the fertile period during which only fertilization occurs. Whether it is clear?
Yes, sir.
Okay. So in some case, the pregnancy will happen. Normally, pregnancy has to happen in the uterus, right? Pregnancy want to happen in the uterus. In sometimes, even in these regions also, pregnancy will happen. Such pregnancies are known as ectopic pregnancies. Such pregnancies are ectopic pregnancies. That is a harmful condition. Ectopic pregnancy, the pregnancy outside the uterus, not inside the uterus, outside the uterus in another region, that is known as ectopic pregnancies. So normally, if you're looking into a uterus, normally if you're looking into the uterus and in this region, this region is known as cervix. In the cervical region, that contains large amount of mucus. The cervix will contain large amount of mucus normally. This mucus, sperms cannot penetrate. The mucus, sperms cannot penetrate to the mucus, sperms cannot penetrate and enter inside. Whenever estrogen is high, whenever the estrogen is high, if in a female, estrogen is high, whenever the amount of estrogen is high, what will happen, that will help in thinning of cervical mucus. Thinning of cervical mucus thinning will happen. So sperm can enter. Thinning of cervical mucus and sperm can enter whenever there is the thinning of mucus. Okay. So from the vagina, from a female's vagina to cervical mucus. Vagina to cervical mucus, to vagina to cervix, the sperm will do flagellar movement only. Sperm will do flagellar movement. A sperm is having flagellar movement only. In uterus, if you're looking inside the uterus, sperm moves by, in the uterus, sperms moves by muscle contraction. Sperm moves by muscle contraction. Muscle contraction will help for the sperm movement. So inside the fallopian tube, inside the fallopian tube, which type of movement, inside the fallopian tube, there is ciliary movement. There is ciliary movement inside the fallopian tube. That ciliary movement will help for the transport of ova. So now we are moving towards the next session, that is fertilization. Fertilization that we are going to discuss. The gametogenesis is over. Whether it is clear, everything is clear. How the sperm is getting produced and how the ovum is getting produced, whether it is clear.
Yes, sir.
So now, if you are looking, you can study about the fertilization. How the fertilization is occurring, how the process of fertilization is occurring. Okay. In fertilization, the first thing is recognition. The first thing is recognition. The sperm want to recognize that it is my ovum. Because if we are looking in an aquatic environment everywhere, different type of sperms are there, different type of ova will be there. First of all, the sperm want to attract or the sperm want to recognize it is my ova. There are 5 events in recognition. The first one is that chemo attraction of sperm. Chemo attraction. That is the first stage. Chemo attraction of sperm. The sperm want to be attracted by chemo attraction of sperm by the egg. Chemo attraction of the sperm by the egg. What egg will do, egg secrete soluble molecule. Egg will secrete soluble molecule. From egg, some chemicals will be secreted out. Some chemicals will be coming out from the ova. Who will recognize that chemical? The moving sperm will recognize that chemical and they will move towards the sperm. This is the first thing. That is a recognition. There is a recognition. Okay. So, then what will happen, Then what will happen next, The second thing, what will happen, The second thing, exocytosis of sperm acrosomal vesicle. Exocytosis of sperm acrosomal vesicle. When it is reached near to the ova, exocytosis of acrosomal vesicle will happen, acrosomal vesicle, and release of enzyme. What will happen? Release of enzyme. The sperm is coming near the ova. When it is reaching near the ova, the sperm's acrosome, very well, a sperm is having acrosome, right? A sperm is having acrosome, and the acrosome is having enzymes and all. What the sperm will do, the sperm will release the acrosome. The acrosome content will be released out. The acrosomal chemical will be released out by the sperm. Sperm exocytosis of acrosomal vesicle will happen. Then what will happen? What is the next stage? What is the next stage? Binding of sperm. This sperm now, binding of sperm. This sperm now will bind to the extracellular matrix. Extracellular matrix of the egg. So above the egg, there are certain membranes, that is vitelline membrane is there, and zona pellucida is there. So over the zona pellucida and vitelline, who will attach, this will come and attach onto the vitelline and zona pellucida. That is the next. Then what will happen next, the sperm passes through extracellular matrix. Through the extracellular matrix, so it is an ovum. Surrounding ovum, zona pellucida will be there, and we know that vitelline membrane will be there, So sperm is attached here now. Then the sperm is moving through all these layers. Then what will happen, Then what will happen next? Then what will happen? There will be the fusion of sperm and egg membrane. Sperm and egg membrane will fuse. Then the sperm membrane, it is reached inside, the sperm membrane and the egg membrane. This is egg, and the coming one is sperm. The sperm membrane and egg membrane will fuse, and this is the first step that will happen during recognition. That is happening during recognition. Whether it is clear?
Yes, sir.
Okay. Okay, so we have to study one by one. That is what will happen during how it is happening, everything we are going to discuss one by one. Okay. So firstly, we can look for the sea urchin. Sea urchin is our model organism. So sea urchin, what will happen? That is external fertilization, right? External fertilization in sea urchin. The fertilization is external. In our case, fertilization is internal. In sea urchin, the fertilization is external. Okay. Who is sea urchin? Sea urchin is a spiny invertebrate that is living in sea. There is a spiny invertebrate. There is a spiny invertebrate. Okay. He is the model organism in DB. Model organism in DB. In developmental biology, we have completed the study about the sea urchin. So sea urchin is our model organism. So what will happen? There is in sea urchin, first of all, species specific sperm attraction. Species specific sperm attraction is the first one. Species specific sperm attraction. Why there is a species specific sperm attraction? You know, this is a mechanism. This is a mechanism to select specific sperm. Mechanism to select specific sperm. We have to select the specific sperm only. So for that, there is a species specific sperm attraction. How it is finding out in an aquatic environment, a lot of different type of ova will be there and different type of sperm will be also there in aquatic environment. How the sperm will recognize, yes, it is my ova. How it is recognizing, you know, ova produce certain ova produce chemo ova secretes chemo attracting ova secrete chemo attracting molecule. Chemo attracting molecule will be secreted by the ova. Chemo attracting molecule will be secreted. That can be recognized by who? That will recognize that can be recognized by the sperm. That will be recognized by the sperm. That will be recognized by the sperm. And you know, it is ova, okay? Near the ova, you know, more amount of that chemical will be there. When it is coming from the ova, that level of chemical will be decreasing. The level of chemical will be releasing. So, you know, it will be high. That chemical will be high in vicinity. High in vicinity. Near the ova, that chemical will be high and less in distant. Less in distant. Anyhow, if one sperm is coming here, that will recognize it and it will move towards the higher concentration region. It will move towards the higher concentration region and eventually that will reach near our sperm. So, you know, that chemo attracting chemical, a chemical is coming, right? That chemo attracting chemical can be called as, sperm activating protein. That can be called as sperm activating protein. We can call it as sperm activating protein or SAPs. Sperm activating proteins or SAPs, we can call it as SAPs. In sea urchin, that chemical is known as, resact. That is a chemical in sea urchin. That contains, that is 44 amino acid length. That is 44 amino acid length protein. That is resact. In purple sea urchin, in sea urchin that is resact. In purple sea urchin, if you are looking for purple sea urchin, you know, the chemical is known as sparact. The chemical is known as sparact. That chemical is known as sparact, okay? That chemical is known as sparact. And here that chemical is known as resact. And its receptors. Receptors are present in receptors are present where the receptors you can see, you know, receptors are present in sperm tail. If you're looking for sperm tail, you know, you can see the receptors for that. So the chemical substances which is coming from the ova, that can be recognized by whom? The receptor that will be present in the sperm tail. This is the chemo attraction. Whether it is clear?
Yes, sir.
Okay. Suppose it is a sperm tail, okay? This is our sperm tail. This is our sperm tail. And this is our sperm tail, okay? This is a sperm tail. Okay, this is a sperm tail. I am drawing a sperm tail here. Okay. Here I am drawing our receptor. Our receptor is here. This one is our receptor. This is a sperm tail and this one is the receptor. Okay, receptor. This is known as a receptor guanylyl cyclase. Receptor guanylyl cyclase. This one is a receptor guanylyl cyclase, or RGC is a receptor present here. So who is coming? Who will be coming? And this one, what is here? This one is a cyclic GMP gated calcium channel. So through the channel, calcium can enter into the sperm, and this is a cyclic GMP gated. Cyclic GMP gated means it requires cyclic GMP to work. So our resac is coming. So from the water, resac is coming. And whenever the resac is coming and resac is binding with its receptor, you know, whenever it is binding with its receptor, what will happen, you know, the sperm will convert GTP, guanosine triphosphate will be converted into CGMP, cyclic guanosine monophosphate. It will be converted into guanosine monophosphate. Whenever it is getting converted into guanosine monophosphate, you know, then this channel will work. Whenever this channel works, you know, whenever this channel is cyclic GMP gated calcium plus channel. So what happens, you know, calcium enters. So it is a calcium channel. Calcium enters from C, from C to tail. From C to tail, calcium will enter. What will happen to the calcium, you know? Calcium activates dynein. Do you remember dynein? Who was dynein? Who was dynein? Where the dynein was present? Anyone? Dynein. Any idea about dynein? Do you heard about the term dynein in before? Just tell me. Do you heard about the term dynein, or whether it is very first for you? Dynein, is it is first for you, or you have already heard about dynein? Come on.
sir.
Huh?
Yes, sir.
Who is dynein? Where you have heard about dynein?
Spermatogenesis.
In spermatogenesis, inside the sperm, dynein was there, right? What was the function of dynein? Anyone? Arul Jyoti, Harini, Sudipti, Subulakshmi. What was the role of dynein? What dynein was doing? You are not studying. You are not studying. You are not studying. You are not studying. We won't go together. Don't think that in the note எல்லாம் வச்சுக்கிட்டு free time கிடைக்குமோ படிக்காம நினைச்சிடாதீங்க, படிச்சுக்கிட்டே இருங்க. So dynein is a motor protein. Dynein is a motor protein. It is a motor protein. That means it helps for movement. It is a motor protein. Okay. So what is happening there in tail? There is calcium activate the dynein. Dynein is a motor protein. So what dynein will do? Dynein will do ATP hydrolysis. Dynein will do ATP hydrolysis. ATP will be broken down. So what will happen? ATP broken down means more amount of energy will come. Whenever more amount of energy will come, what will happen? You know, flagellar movement at more amount of ATP is coming. Flagellar movement will happen. Flagellar movement at higher speed. In higher speed, you know, flagellar movement will happen whenever there is ATP hydrolysis. This is happening in chemo attraction, in chemical substance. When it is coming towards the, when a sperm is recognized in the chemical, this phenomena is happening. Whether it is clear? Everything is clear for you, but you are not studying. We can only teach. Studying is your responsibility. Again, I am saying the same thing. Studying is your responsibility. Okay. Okay. I think it's enough for today.